it will be required for the other part of the body. Now here you will be learning that in liver and muscle, both places glucose 6-phosphate can be converted into glucose. In muscle, glucose is immediately utilized within the muscle, whereas in the liver, it is taking care of the rest part of the body. So whenever I'm saying glucose 6-phosphate to glucose, that means it could be for brain, it could be for other tissue where we require glucose urgently. What else? Glucose 6-phosphate can also enter in another pathway which you have learned in the last class, which is a pentose phosphate pathway, where it can produce ribose and NADPH. Both are required for the biosynthetic reaction. Ribose can be required for the synthesis of your nucleic acid. NADPH is required for the other synthesis where reductive biosynthesis are involved. Now, looking at glycogen metabolism is not so simple. Glucose is being removed from the glycogen. And if you recall, glycogen is a molecule which is having branch. Now, how does the branch form? Branch formed by alpha-1,6 linkage. So when I'm talking about this reaction, which is a phosphorylase reaction where phosphorylatic cleavage of glycogen occurs to produce glucose 6-phosphate, and the rest part of the glycogen is left, as I said in the previous slide. This reaction is a very interesting reaction. It is cleaving the glucose 1-phosphate from the terminal residue. And this we have talked when we were in the first few slides where we were talking about the carbohydrate. Now you may have a question, why glycogen is not converted into glucose? Why in glucose 1-phosphate? Is there any advantage in the metabolism? Answer is yes, because here you don't require energy ATP to convert glucose into glucose 1-phosphate. So once glycogen is metabolized, still it is an energy rich molecule. So glucose 1-phosphate to 6-phosphate conversion does not require ATP. So whenever the cell requires this for glycolysis, or for example, it is required for even gluconeogenesis, or it is required for this pentose phosphate pathway, it does not require for the ATP, and that's why this reaction is a phosphorylatic reaction. It is not common reaction. It is not the reaction where you see the breakdown is happening in the help of water. So this is a self-driven reaction where phosphate itself makes a molecule which is an energy-rich molecule. So this is the reaction. This is the showing you the structure of the molecule. Now, in the next reaction, conversion of glycogen to glucose does not happen just with the glycogen phosphorylase. Why? Because there is a branching which is present. So what happens from the branch? The cutting of the branch happens with the help of glucosidase, which is alpha-1,6 glucosidase. This is the separate enzyme which is cleaving the bond, because this is a different type of bond. It is alpha-1,6 bond. Initially, phosphorylase for phosphorylatic activities cleaving alpha-1,4 bond. Now this is alpha-1,6 bond. That's why you require a different enzyme, and then you can produce. Other than that, when this branch comes to the end and there is only 3 residue which is present, then it is transferred from one part of the branch to another part of the branch, and that's why you require transferase. So conversion of glycogen to glucose requires 3 enzymes. One enzyme is phosphorylase enzyme. Second enzyme is the one which is this is the first enzyme is a phosphorylase, which is converting glycogen into glucose 1-phosphate. Second enzyme here, alpha-1,6 glucosidase, and third enzyme is the one where they transfer it from one unit to another unit of the branch, and that is the transferase. So this is the transferase enzyme. That's what I have talked about. So once you have glucose 1,6-phosphate, as I said, they can be converted into glucose 6-phosphate by enzyme phosphoglucomutase, and that phosphoglucomutase that shifts phosphoryl group from first position to sixth position. And this is a similar reaction you have seen in glycolysis also, so I'm not repeating that. Now, phosphorylase is regulated by several allosteric effectors. Now, look at the reaction. Glycose is again converted glycogen, it is a futile cycle. Energy will be wasting, cell will be wasting energy. So these reactions are controlled by the enzyme. And these enzymes are in turn controlled by hormones. So what are the hormones? So there is a reversible phosphorylation reaction which will control the phosphorylase enzyme. And these reactions are controlled by hormones like insulin, epinephrine, and glucagon. The difference in control of glycogen metabolism in two tissues, skeleton, muscle, and liver cell, they are different. As I said, that glucose produce energy in the muscle for itself, whereas in the liver, it is for whole organism or whole body. Now, this is a very typical reaction which shows how phosphorylase B enzyme can be in active state or inactive state. Of course, you will be further looking at another way of controlling, but this is one way by which glucose can be, phosphorylase B can be controlled. And what it does is that if there is a lot of AMP shows low energy state, so it becomes in relaxed state. That means it should produce more your glucose. On the other hand, if there is a more ATP, no production of glucose is required, and they will be in the tight state, or the one which is not involved in the conversion. So this is what is happening in the muscle. In general, hormone they regulate, they convert inactive phosphorylase kinase to fully active phosphorylase kinase, and once phosphorylase kinase is active, it can convert inactive phosphorylase B into phosphorylase A. So all these reactions are, I'm going to discuss about that. They are reactions which are happening one after the other, and they are several controlled. So this enzyme is controlled by the hormone. There is an enzyme which is called as a pKa. pKa is protein kinase A. I'll talk about that. Then it also requires calcium. Once both of them are there, they can phosphorylate. There's a beta subunit which is phosphorylated, and calcium is attached to the delta subunit. Once these two are attached, the enzyme becomes fully active. Otherwise, even just by phosphorylation or just by calcium attachment, they are partially active. And once they are active, they can convert phosphorylase B to phosphorylase A. So you have a mechanism where you can make them fully active. That means reaction is going to be very fast. You have a mechanism where it can be partially active. So that way, this enzyme is controlling the reaction. Now, these are written thing and too much, in writing. I will explain it to you in the actual, mechanism, how it is happening. It is a kind of a cascade. So what happens? The signal molecule is the hormone, which is epinephrine or glucagon. They will bind to a specific receptor. Epinephrine bind to 7 transmembrane receptor, whereas a glucagon binds with the glucagon receptor. And then binding activates a protein, which is a GS protein. S refers to stimulating protein. It is a G protein. It is a stimulating protein and stimulates G protein. And once G protein gets activated, it activates another enzyme, adenylate cyclase. I will show it to you. It is a transmembrane protein. It catalyzes formation of cyclic AMP from ATP. So ATP is present in the cell. It will form cyclic AMP, and the cyclic AMP level activates protein kinase. So you see, this is not one reaction, several reactions, one after the other. One activates, this will pass on the signal to another one. And that's the way signal transduction occurs. So in this cascade, let me just show you. So epinephrine, here this is epinephrine, it could be glucagon also. So epinephrine binds with this beta adrenergic receptor, whereas a glucagon will bind to another receptor. But both of them, what they will do is that they will activate this molecule. This is the G protein, and this G protein, once it is activated, so what will happen? It gets activated, G protein will activate it, and they will remove these two alpha, here it is made up of 3 subunits. Its beta and gamma subunits get dissociated, and alpha subunit helps in the transfer of GDP to GTP. And once GTP binds, this becomes active along with the adenylate cyclase. Now the function is of adenylate cyclase. So far, adenylate cyclase was inactive. Once G protein binds, it becomes active. And what it does is that it converts ATP into cyclic AMP. Cyclic AMP binds with the protein kinase A. A is referring to AMP. So once A is activated, this green one is active form. This will activate phosphorylase kinase into phosphorylase kinase active form. And then phosphorylase B enzyme becomes phosphorylase A, which is active form. Once this active form is formed, then it will convert the this will be the one which will be activating phosphorylase kinase, and once activated phosphorylase A is there, it will convert glycogen into glucose phosphate, and this whole reaction is being controlled. Now I have shown you two set of reaction, one on the right hand side, another on the left hand side. Both you have a box. Up to the box, the reaction is same. There is no difference. Epinephrine comes, binds, and finally, after a cascade of reaction, it activates protein kinase here, A here, protein kinase A here. Now, protein kinase has a dual role. On one hand, it activates phosphorylase kinase, which becomes active phosphorylase kinase. And that's why it converts phosphorylase B to phosphorylase A, and phosphorylase A will convert glycogen into your glucose molecule. So from glycogen to glucose 1-phosphate, you will get. But on the other hand, the same protein kinase A inhibits glycogen synthase. So it will make glycogen synthase in inactive form. So red one shows inactive one, and that will not convert glucose to glycogen. So even if you have glucose 6-phosphate, which has been found from glucose 1-phosphate, they will not synthesize glycogen. So same enzyme controlling these two reactions, and thereby it will be stopping on one hand, it will make more so on one hand it controls one reaction, on the other hand it controls the other reaction. Now tell me if there is any thing you require any explanation. If there is no explanation required, you can go through. These are very simple reactions. It does not require much complexity. I will take 4-5 minutes more to explain you another metabolism, which is an interlinked metabolism, where I have already talked about how from fat how we utilize the one of the part, and that was glycerol part. So fat, when they are metabolized, first thing is let's see what is the fatty acid metabolism. I'm regulating fatty acid metabolism because most of the triacylglycerol, they will be converted into fatty acid, and on the other hand they will be glycerol. And glycerol metabolism you have seen in the first part of the lecture. Now here what we are going to talk about is that how fatty acid is being metabolized. So fatty acid has a different role. On one hand, it is a building block of the phospholipid and glycolipid, which you already know, which is a part of the membrane. They can be, if fatty acid is attached to any of the protein, then they can attach to the membrane because that becomes hydrophobic. Third one is a fatty acid is a fuel molecule. So we will be focusing on this part, that how fatty acid acts as a fuel molecule. And fourth one is a fatty acid derivatives such as the hormones and intracellular messenger. So some of the fatty acid, basically long chain and having double bonds, they are derived into hormones and mostly prostaglandin. I'm not going to cover in my class, but the prostaglandins are synthesized from the fatty acids. So they are the one which are intracellular messenger. They are being synthesized from the fatty acids. So most lipids, when you digest, is in the form of triacylglycerol. So suppose oil, whichever we are consuming, that is nothing but triacylglycerol. So what happens to this triacylglycerol? They are degraded into fatty acid, and they are being absorbed by intestinal epithelium. So our body, it goes and it is absorbed by our intestine. Now TG or triacylglycerol in intestinal lumen are incorporated into micelle, and you know that if micelle is not formed, they will not be absorbed because they are they have to be converted into micellar form. And then there are pancreatic lipase inside the our digestive system, and this lipase will degrade it. And then the micelle formation occurs with the help of bile salt. So this bile salt plays an important role in the metabolism of the fat. If it is absent, what will happen? Most of the fat will be excreted from the body in the form of feces, and that is what is a disease, and its condition is referred as steatorrhea, because steato means fat. So fat is being non-metabolized and it is excreted. Now lipase, what it does is that lipase digests triacylglycerol into free fatty acid, and monoacylglycerol or glycerol, and the digestion product is being absorbed in the body. So TG, you have a lipase, it will cut this bond, the ester bond, and your fatty acids are being released. So this fatty acid metabolism I'm going to talk about in few minutes. Now here in lumen, inside the intestine, what is happening? TG is converting to fatty acid in presence of enzyme lipase. Fatty acid and monoacylglycerol, they enter, and this is also a very typical process. You need a real transporter, you need something to carry the fatty acid, because otherwise it is a charged one, it cannot enter inside the cell. So there is a mechanism where carnitine is the one which binds to the fatty acid, and then they enter. Once triacylglycerol enters, either it is further metabolized and your fatty acid is again used for the process, or this fatty acid directly which has entered can be used. Alternatively, it can incorporate with the lipids and protein and can do other work and forming chylomicron. But on the other hand, this fatty acid which has entered, which is not binding with monoacylglycerol, they can be further metabolized, they can produce energy. So how it produces energy? The fatty acid is present, it is a charge, it will not enter in the metabolism unless it is activated. So there's a process of activation, and activation requires coenzyme A. So coenzyme A is added in the fatty acid and it makes fatty acyl CoA, and this happens in presence of ATP. So this is an energy requiring process. As you have learned in glycolysis, for a few steps require energy or ATP for making the molecule to work further, and the same way here the coenzyme addition is required for further metabolism. Now here, if I look at, I see just one ATP which is driving the reaction. But actually what happens, this reaction happens in two steps. First, fatty acid is converted into adenylate acyl adenylate. That means AMP is added and PPI is removed. Second step, AMP and acyl CoA comes and makes your acyl CoA. And this acyl CoA, when formed, this PPI, both these reaction, this one reaction and two reaction, both are reversible reaction. And this reaction, in one hand you are synthesizing energy, you are breaking energy rich molecule, on the other hand you are making energy rich molecule. So the reaction can be in equilibrium. But what happens to derive this reaction in forward direction? The energy is taken from PPI. This PPI is broken down into 2 orthophosphate, 2PI, and that's the way reaction moves in the forward direction. So this is the typical reaction. At the end, your fatty acid R could be any number. So it could be 18 carbon or any carbon, and which is activated, making into fatty acyl CoA. AMP is formed, and then so this reaction in some book it says that this requires 1 molecule of ATP. Some places in some of the books it counts the breaking of the phosphoryl this PPI, and that is why conversion of PPI to PI also is considered as 1 molecule of energy. That means ATP. So in some books you will find when energy calculation is done, it requires 2 ATP molecule or equivalent to 2 ATP molecule. Now I will give you, not in detail but in a very simple way, that once acyl CoA is formed, R is the number. Suppose I start with 18 carbon or 16 carbon. So what will happen at the end? That is what we are going to discuss. So the first step is oxidation, where you have a dehydrogenase. FAD is converted into FADH2, and you know that FADH2 can produce energy. On the next step, it is a hydration reaction. Water enters, and this double bond is getting saturated. It is preparing the molecule for to be metabolized. And next step is again oxidation. So first step is oxidation, third step is oxidation. So this reaction typically moves with a cycle of oxidation, hydration, oxidation, and third is a thiolysis. Thiolysis is that the bond is cleaved at thiol group. So first reaction is which I'm not considering, which is the activation reaction. After activation, there are 4 set of reaction. Why I'm talking about this 4 step of reaction is that these 4 step reaction, they keep on repeating till the molecule of fatty acid is broken down into 2 carbon acetyl CoA. Let us see. So suppose I start with acyl CoA, it is converted into trans-enoyl CoA, and then to 3 hydroxy acyl CoA, and then again oxidation happens, and then last one is coenzyme A comes, and thiolysis occurs. That means you get at the end 1,2 carbon molecule, which is acetyl-CoA. Of course, what is the remaining one? Suppose I start with the 16 carbon, I end up with this 2 carbon, and this will be 16 carbon. Now this 16 carbon again re-enter in the same step of reaction. So it will keep on repeating. So if I start with the 16 carbon, how many cycles should happen? Yeah, any one of you? So here the point is that at the end, when 2 carbon is left, no more cycle happens. So if I start with the 16 carbon, only the number of reaction will be 7 cycle instead of 8 cycle, because 8 cycle will not be required, as once you get all of them converted into acetyl-CoA. So what I'm trying to say here is that each cycle you get 1 molecule of FADH2, 1 molecule of NADH. So depending on the number of acetyl-CoA, this chain, you will produce these number of NADH and FADH2, which can again produce energy. And at the end, when you get this acetyl-CoA can enter in the citric acid cycle and can produce energy. To sum up, I have given all the reaction, all the enzymes which are involved in these steps, and ultimately what you get is that you get the fat which is being metabolized. Fatty acid is converted into their individual acetyl-CoA, and these acetyl-CoA can enter in the TCA cycle to produce energy. Any questions so far? Any question from any one of you?
Sir, what about the quiz?
Quiz will be, I will write to all of you. Quiz I will take on 4th in the evening. I will announce the time, whatever is comfortable to all of you. 7 o'clock or 8 o'clock, is it okay for you?
Yes, sir.
Yes, sir.
So I will write a mail to
No,
you
sir.
giving you
We
all the
already have a
instructions.
quiz at that time.
So on 4th, you will have the normal class. And in the evening, what I will do is that I will have 20 minutes, or that means half an hour. There will be quiz. Followed by quiz, I will give you open, time, maybe 1 hour, where you can discuss any doubts, any question, any difficulty in your this course. So that 1 hour will be, so you keep almost 1.5 hours free for you, half an hour for quiz, and then we'll connect on the Microsoft team. We'll discuss any question which you have, any doubt. Okay.
Sir, when will the class happen on 4th? Because it is a Monday.
Sorry, then you have a class on 5th. Do you have a class on 5th?
No, sir,
You
5th
No,
is
sir.
holiday, sir. No class day because 6th is exam.
Hmm. Okay. See, I don't know. The timetable is such that we cannot. So 4th, do you have the time in the morning hour? I thought that we have a class.
Yes, sir, I think we have time in the morning.
So let us put it in the same timing wherever we have
Sir,
normal class. Yeah.
sir, 9:30 to 11 we used to have CLL,
Hmm.
and now that is over, so you can take it in between 9:30 to 11.
Okay, that is fine. So I'll on Monday 9:30 to 11. What I'll do is that I have just half an hour class, and then I'll give you half an hour for your discussion. If you have something, or 15-20 minutes for discussion. I want more discussion from your side. I want if there is any difficulty from any part of the course, so that we can discuss it out. And because so far, I think after your first minor, where you have the difficulty. After your first quiz, what is the difficulty. So if you have any question related with your course, I'll be open to in the morning as well as in the evening when we have your quiz. So 4th, we are having one class from 9:30 onwards. 9:30 to 10, I will have a class. There, I'm just going to brief up about the signal transduction, because that is a part of your course. And in the evening, what I'll do is that in the evening, we will have a quiz, and followed by the quiz, you will have again half an hour slot where you can discuss any question if somebody has missed in the morning. So 9:30 to 10:30 in the morning, and say I'll just look at my timetable. 7:30 or 8 we will start quiz, and for you keep 1 hour approximately for with you. Okay. So with this, I will wish you very happy New Year, because we are starting now in New Year.
So, excuse me,
Yeah.
sir. I guess some people have a certain course which has a quiz like around 7 on 4th.
So can we have at 8 o'clock?
I guess so,
Give me
yes.
some time. So 8 o'clock, I will not start at 7. I will start at 8. So I'll circulate this. I do not want anybody to miss it, because it is going to be 10% of your overall evaluation process. So everybody should get chance. So I will not restrict it, because it is not on their lecture hour. So on 4th, we will have morning class 9:30 to 10:30 with the discussion. And in the evening, we will have a quiz plus your, any doubts that can be clear. Okay, then I will stop at this point.
Sir,
Thank
so
you very
quiz will
much. So
be in the
I'll
morning on 4th.
the quiz in the evening. Quiz is in the evening. Or if you want, I can have in the morning also. I don't
So
have any
I guess
problem.
evening is better, 8 to 8:30 or something.
Ah, yes, sir.
So, in the evening, I will have, as you said, that 8 to 8:30. If you are comfortable, I'll have in the evening itself. So I'll announce this. I'll circulate the mail today itself about the class as well as your quiz and any doubt clearing session. Okay. So
Okay, thank you, sir.
thank you. Thank you. I'll
Thank you, sir.
stop at this point. Thank you very much.